Okay, so good morning. We'll continue from yesterday's class. Today, we'll discuss the third epigenetic player. The first epigenetic player is a DNA methylation or nucleic acid methylation. The second is histone modification, which influences the gene expressions, which you can connect. See, DNA modification, obviously, you can think it can modify the gene expressions. Histone modification, it can influence the associated gene regulations. The lncRNA, it's a recently identified epigenetic regulators. It also modifies and play a very important role in bringing the epigenetic changes in the cells. Initially, it was lncRNA, that is long non-coding RNA, and later many other regulatory RNA, small RNA also was identified, and it's a role in the regulating the epigenetic players. Let's look at briefly what is long non-coding RNAs. The RNA molecules more than 200 or longer than the 200 nucleotides that are not translated into the functional proteins, which you call or define as a long non-coding RNAs. It should be at least 200 nucleotides. There are many genes, almost as per the annotations, 16,000 lncRNA coding genes has been annotated. But however, the transcriptome data has identified there are more than 1 lakh long non-coding RNA has been so far identified in the transcriptome data. So that means only 16 or 20,000 genes maximum, but many are produced due to the splicing and so on. Like mRNA, these long non-coding are also capped with methyl guanosine at 5' end. And also this gets polyadenylated at the 3' ends, almost similar to the mRNA modifications. And it has its own promoter to produce the lncRNA, long non-coding RNA. But however, some of the lncRNA produced from these enhancer regions and also promoter regions. I will show you briefly what is enhanced RNA. The RNA exclusively produced from the enhancer regions, which participate in the enhancer function as well. So, likewise, there are many long non-coding RNAs has been identified in the human genome. What are the major functions in connections with the epigenetics as well? One of the prime function of long non-coding RNA, it regulates chromatin structure. Shaping of the chromosome also has been regulated. The best example is X-chromosome that we will discuss today. X-chromosome inactivation is tightly regulated by these long non-coding RNAs. And it also regulates various transcriptions, coding genes as well, the long non-coding RNA. And it play a role in nuclear organizations, sequestering the heterochromatin regions in the specific loci or anchoring the repressive regions in the nuclear periphery and so on. And also it has a post-transcription functions. So these are the key functions about these long non-coding RNAs. So how is been synthesized? So how long non-coding RNA synthesized in the cells? This is the mRNA synthesis pathway, as all you know, and the lncRNA also follows the similar pattern. But there is one difference is the lncRNAs tend to retain within the nucleus or chromosome. That's called nuclear retentions. Some are actually exported for its function in the cytoplasms, but majority need to retain because that is a place it has a major role. So that mechanism is called nuclear retentions. Like mRNA, this also gets splicing, undergoes splicing, and then form mature lncRNA. It also gets capping, 5' capping, and 3' polyethyling as well, but does not encode for a proteins. This is a non-coding RNA. So why it need to retain within the nucleus? The reason there are a lot of mechanisms for this. The main reason obviously to shape up the the chromosome expressions. For that purpose, I mean the chromosomal specific regulation and gene expressions. For that purpose, it has to return in the lncRNA. The main mechanisms through which it get returned through these small nuclear ribonucleoproteins. So this is one of the mechanisms through which it form a complex, and then this has an association with various Pol two enzymes complexes, and then it get tethered. And then because of this weak or poor splicing signal, some of the link, not always, some of the lncRNA has this the region called polypyrimidine track. So this is an important region which regulates its splicing, and because of these having these specific sequences and poor splicing, and then it get returned within the nucleus. So these are the few mechanisms through which it return. The main reason why it has to return within the nucleus is to function within the nucleus. That means chromosome shaping, X-chromosome inactivation, and so on. Right. So how exactly this lncRNA mediates gene silencing? Coming to the epigenetic role, I'll give a few examples. For example, it's a role in a 6 acting function, trans-acting functions. In case of 6 acting functions, the example is I'm trying to explain using this enhanced RNA. See, the enhancer RNA nothing but RNA transcribed from the enhancer regions, and that does not encode for a proteins, that is called eRNA. So what is the main role of enhancer is it activates transcription, it stimulates the transcription, right, enhances the gene expressions. How does it enhance the gene expressions? Primarily forms a looping. So the enhancer regions, it may be near to the promoter or upstream, downstream, maybe even 3,000-4,000, even 10,000 base pair away from the target genes. It may be located anywhere, but still it can regulate. How does it regulate? The mechanism, it's called chromatin looping. So the regions can come closer, and then this can stimulate this gene A. That is directly it can does. Other way around, by producing the enhanced RNA. Without the enhanced RNA also it can stimulate, keep in mind. But in this case through enhanced RNA. How does enhanced RNA does it up? Enhanced RNA forms the special triple helical structures on the enhancer regions, and then which recruit a protein, activate a protein here, and then this protein binds the promoter, and then along with the transcription factor will lead to the activation. So this is the one of the mechanisms through which lncRNA function as a for gene expressions mechanisms. The trans, which I'll explain, it also play a role in the X-chromosome inactivations. That means the RNA produced from the X chromosomes and regulate on the other X chromosomes, distal regions or other genes. So this also I'll explain coming to slides. The other mechanisms through which, I mean, it function as an gene silencing, it recruit the protein. This is the main primary functions. The lncRNA, how does it mediate gene silencing and epigenetic function? These two are rare examples what I explained, 1 and 2, and of course very specific in this case. General epigenetic functions, this one, the C, targeting or recruitment of epigenetic machinery. So what does it do? It binds with the epigenetic proteins. It can be a DNA methyltransferase, it can be a histone methyltransferases, and then it target to the specific genomic loss, basically targeting mechanisms. So this is one of the functions. In this case, an example with X chromosome inactivation, the specific transcript called ZIST play a role using this mechanism. It recruits histone modifying complexes. The another example where it can function as a decoy. So what does it do? It can bind with some of the machinery, some proteins, okay, and then it can sequester that are not available for the gene activation repressions. That's called decoying. This also helps for you few examples. So these are the primary mechanisms, either recruiting or make sure it's not available for the gene activations. So this is how the eRNA functions, that is enhancer RNA functions. So you can see these enhancer regions and then promoter regions. Through chromatin doping, these regions comes closer, and then the gene activation is done by this enhancer RNA. So the enhancer RNA function as a mediator between these two. So this is the mechanisms through which eRNA functions. Keep in mind, without eRNA also enhancer elements can functions. Okay, so you see lncRNA participate in the DNA methylations, chromatin modifications, X-chromosome activation that we will see in today's class. It also been shown with the paramutations and then imprinting. It play at various level in connection with the epigenetics. What I explained the general gene expression mechanisms, but in epigenetics regulation level, it participate at various level. One of the well studied mechanisms is X-chromosome inactivations. This one. When there is a defect in the expressions or maturation of the lncRNA, even defect in these nuclear retention, it will have a various complex diseases. So that means the expression and regulation of the lncRNAs play very important role, like your coding genes. If there is any defect in these also, it contributes at various disease development. Right. So this is one of the beautiful example in terms of recruitments. So where the hot tip is one of the well studied lncRNA. So this is basically OX specific transcript at the distal tip, which is produced from these regions, and then it has an ability to bind with the WDR repeat. You all know this is a docking protein docking regions where the many activator complex will be recruited, and also MLL. Basically, this lncRNA recruits gene activator complexes, and then it recruit to the different nucleosomes by through the looping and introduces the trimethylation marker at the S3K4. So this is one mechanism through which it can regulate, basically targeting. The other mechanisms, again I will come to this. This is one of the rare example. Again, I will come to the specific targeting mechanisms when I explain about the X-chromosome activations. The decoy, so which I explained earlier. So what does it do? It basically binds with the one of the epigenetic machinery, and then the binding makes sure not available for the chromatin modifications. See, for a SIT2, it is a well-known deacetylase, NAD dependent deacetylase, which I explained. Okay, the SIT enzyme, SIT2 enzymes. So in this case, in the pluripotency specific genes, which is always expressed, and then due to this acetylation of these positions, and then the SIT now to deacetylate and make it repressed, right? So these specific lncRNA, when it is expressed, so this is a p53 regulated embryonic stem cell specific lncRNA. That's why it's named as a link PRESS1. When this specific lncRNA expressed, it has high affinity to this protein SIT6. When it binds, it's not targeting. Basically, it is sequesters. It's not make sure the protein is not available to deacetylate. So in the presence of lncRNA, it is gene activations. In the absence of lncRNA, it is a gene repressions, because it can go and deacetylate, because it is a H-tag, right? So you see how the lncRNA can contribute the differentiate gene expression in the absence and presence on the single loci. In this case, well studied with using the pluripotency genes, because when they deplete this lncRNA using the knockdown strategies, they could see down regulation of some of the pluripotency genes, which clearly identified, yes, the lncRNA can regulate the protein availability, the H-tag availability on the specific genomic loci. right. The one more example, targeting examples. This you can connect in the early days of DNA demethylations. If you recollect one of the DNA demethylation classes, I explained about the proteins that's called GAD, that is a growth arrest DNA damage inducible alpha. Okay, GAD 45A protein. Okay, so there is a specific link RNA called TARID. Okay, so this TARID is basically TCF21 antisense RNA inducing demethylations. Okay, so RNA inducing demethylations, specific link RNA. What does it do? It forms a R loop. Majority of the targeting link RNA forms the R loop. See, R loop is nothing but triple helical structures where you have the double stranded DNA, okay, and then link RNA paired with one of the strands. So this is due to the link RNA binding, and then these two are pairing, these two are slightly away. Okay, this is triple helical, and this is you define as a R loop. So this is the definition of R loop, the characteristic of the R loop. It has a lot of function in the cell. In our chromosome, we do have R loop because of the link RNA RNA interactions to the specific DNA. So here the TAR edit, that is a TCF21 antisense RNA inducing demethylations, it binds to the specific loci, and then this RNA, when this forms this structure, it is unique actually. See, in other cases what I explained, link RNA directly recruits, but here link RNA it recruits by forming the R loop. That is the difference. Okay, it doesn't recruit in this place, right? Upon interactions, it has a special structure, and that recruits the protein called GAD45A, and then that has an affinity for the thymine decarboxylase and then TED proteins. So this is the different. This is also recruitment, but different mechanisms. Whereas this recruitment is a different mechanism. It doesn't form R loop, but it recruits the proteins, and the protein can assemble to introduce a methylation. But here the recruitment itself is specific to the R loop, which is selectively found by this link RNA. That means this sequence also very important. It's not only just random R loops, the TARID specific R loop, that's very important. Okay, so once it assembles these complexes, it induces a DNA demethylation, because TDG, you all know what is the functions. It takes care of this TET mediated final accession pathway, and TET obviously induces the DNA demethylation, which you all know. Before the discovery of the TET, a few of the studies believed this is the protein actually induces the DNA demethylations. The reason they thought it recruits TDG instead of going to the oxidation mechanisms, hydroxy, formyl, carbyl, it can directly cleave the methylated cytosine and can introduce the new cytosine. This was the initial active DNA methylation mechanism because of the protein, this protein. But later only they identified not only this protein, there is one more protein, intermediate protein, which function as the oxidation mechanisms. Okay. So this is the one of the mechanisms through which the link RNA can regulate the epigenetic functions. Okay. So let's move on to these specific examples, which I'll highlight, because the contribution of link RNA in epigenetic phenomena are well characterized with the X-chromosome inactivations. It's one of the famous epigenetic phenomena that we discussed. In fact, the concept about the X-chromosome inactivations discussed much earlier. In fact, the Mary Line proposed in 1961. But keep in mind, this team proposed, yes, there is one of the X-chromosome in a female gets inactivated, but the scientist called Barr, okay, so and his team, they were the one first identified in a female cell there is always a condensed nuclei. That is why it's called bare body. The inactive chromosome is called bare body because the structure was discovered by the scientist. So it was in 1950, almost 1949 to 1950s time. So those time itself. But the concept of inactivation started almost after 10 years, but it was not very clear which chromosomes, whether mother specific, father chromosome. Even till now, it is the mechanisms. It's not well established. Most of the information what we available using the mouse model, which always we think it's matching to the human model as well. So the X chromosome inactivations, one of the in females, it happens in the cells during the embryogenesis, which I'll show you how does it exactly happens. Once it inactivated, and particular some chromosomes, it's inactivated throughout the development. So whatever the cell derived from that particular stem cells, in that cell will always have an inactivations. So this is defined as a bare body, and then it's only in a female, and then the rare chromosomal abnormality also the X chromosome inactivation takes place. Even the female, if it has a more than 3 chromosome, X chromosome also, it keeps only 1 and 2 gets inactivated. So this is what happens. So this is the developmental picture. From this forced fertilization, this is a zygote and 2 cell, and then 4 cell and morula. And during this stage also inactivation takes place, but only father chromosomes. Okay, so that is why it's called imprinted inactivation. Only the father specific chromosomes or paternal chromosome inactivated during this stage. I'll show you the cycle and understand how exactly this inactivation mechanism takes place. Then it start reactivation during this phase, and then when the embryonic day 5, just before implantation time. So this is early pre-implantation embryo. You call from zygote, from forced fertilization to prior implantation, you call pre-implantation embryo. During that time, only father chromosome in these cells are inactivated. Then both are getting reactivated, and when it is in a morula stage, I mean, after near to at the time of implantations, either mother or father chromosome, one of the chromosome in these cells gets inactivated, and then from there only it is inactivation continues through the development cycle. So you see, you just follow what I explained. So this is a very complex cycle. This gonad cell, either sperm cell or egg cell, will have the active X chromosomes. Okay, so the X chromosome in these are active. That means not repressed. And post fertilization also, in a zygote level, it is active. Okay, and then when it is developing towards morula stage, 8 cell stage, and next level, you see the paternal chromosomes gets inactivated. So that's what I'm trying to say. From here, it start inactivating one of the paternal chromosomes. Then when it develops towards the after implantations, there is a 3 level of differentiation, you all know. So this is called blastocyst stage, and then there is another stage called tropoectoderm. This is a tropoectoderm only will develop into the placenta, and then the blastocyst, which has the inner cell mass ICM, and this is the one which develop into the whole organisms, right? So this is the basics. Now here the father chromosome, that's the paternal chromosomes, get inactivated. Then there is short reactivation in this phase. Okay, so the activations get erased from the father chromosomes. Then somehow the placenta alone carry the father inactivations. That's father specific chromosome. So the placenta chromosomes will always carry the father specific inactivations. That has been identified. That is the paternal inactivations. And also some of the primitive endoderm, which is not well characterized, but at least the placenta is well documented now. So once that is continues, then after reactivations, then this is the place the random inactivation takes place. You have the a bunch of the stem cells, right, and that place each and every cell undergo differential one chromosomes, but need not to be a same. Every cell have the same whether father or mother. You have the 100 cell means 100 cell. You will have a each and every cell have a different. One cell may be father, one cell may be a mother. So that's random inactivation. It takes place. Moment it completed the inactivations, then whatever the chromosome inactivated from this stem cell, and then if that developed into bone, blood, whatever tissue derived from the stem cell, inner cell mass, that will carry the inactivation. So this is how the inactivation cycle takes place in the female, the X chromosome inactivation. You see, the early stage inactivation only father specific, and then later it gets reactivated, and then during the inner cell mass after implantation, then the random inactivation happening, and that continues. And one of the mechanisms, what is a factor contribute the selection, random selections, and that is when unknown. If that doesn't happen, you have the failure. The abortion all happens due to this case also. When this inactivation is essential, it is not happening, the baby will not develop. So that's very clear. This X chromosome inactivation is essential for the normal development. And before that, I move on to these mammalian inactivations. I'll introduce. We will talk about the X chromosome inactivation separately. The three model organisms are well characterized. One is mammal, and second Drosophila, and third C. elegans. Mammal, why X chromosome inactivation needed? Dosage compensations. So the male has a X and Y, and Y obviously is smaller the chromosome, less number of genes, and X has the more chromosomes, so more genes. Sorry. And a female 2X, that means more genes, right? So what happens? You have two alleles, means from both the alleles you have the equal expressions. If you have two chromosomes, basically you have 4 alleles, and then you have the double the gene expression, which is not needed for our systems. That is why female evolved to shut down or suppress one of the X chromosomes. So basically, for achieve the dosage compensation only, the mammalian systems developed to suppress one of the X chromosomes in the female. In Drosophila, it's quite opposite. In here, the female takes care of it. In Drosophila, the male fly, there the male fly produces more to have dosage compensations. The female keep producing more genes in the fly, whereas in a Drosophila, instead of shutting down one of the chromosomes, the male fly produces double the expression so that you have the equal development. In C. elegans, quite different, because it's anyway in hermaphrodite, right? It has these both male and female in these single worms. Also, it has a male worm separately. The male worm will have the hermaphrodite will have the XX chromosomes. That means both sex organs present in the single animal, and then male will have XO. Here it's very interesting. So when I talk about model organism, I'll briefly explain. Here it shut down double the time. So in one case, it just shut down half gene expression. Here doubles the expressions. Here double the suppression. So this is how the X chromosome inactivations is carried out to have the dosage compensation of gene expressions. Yeah, so this is the concept I'm trying to explain. These why the dosage why these X chromosome inactivations needed in a female to achieve the dosage compensations. Right. So the dosage, because of the dosage compensations, you have the the phenotypes are in mosaics in nature, but in human we don't have much of this any coloring color producing genes in our X chromosomes in the females. So that's why we don't have the phenotypes. Whereas some of the animals which I show you, which produces the different color due to the X chromosome inactivations. So this is genetically mosaics. The reasons, the random inactivation. As I said, it's not uniform in every cell. One cell father, one cell mother. Because of these, we have this. That means female are genetically mosaics. Female chromosome, X chromosomes are genetically mosaics because of the random chromosome inactivations. Right. The primary epigenetic players which mediates the X chromosome inactivation is the link RNA called ZIST. Okay. The ZIST, it is link RNA, one of the longest long non-coding RNA so far studied. You see, it is nearly 17 kb length RNA molecule, which is very long. It is produced from the center called XIC. You see, you have the two X chromosome in a females. One is always active, one is always repressed. X active, X inactive. The inactive means it is not completely inactive. There is a center called XIC, X chromosome activation center. From here, it produces the link RNA. So this link RNA called ZIST, and this will take care of suppression of these whole chromosomes. So this is called cis acting. But the RNA which is produced from here, that's another example I said, trans acting. If you recollect a few slides before I said. So there is RNA which is produced from here, which basically sequesters, not allowing for the acting other. So through decoying, which I'll also show you. So this also classified under these transacting link RNA, which I'll show you. That's two different RNA which is produced from these both active and inactive chromosomes. Okay, so one important RNA which you will recollect is ZIST. ZIST is X inactive specific transcript. X inactive specific transcript. The RNA is produced, ZIST RNA, and then what does it do? It just spreads on the own chromosomes and makes sure repressed. So while spreading, what happened? It recruits the epigenetic machinery. And you can see in a XX female, one bare body. In these rare chromosomal abnormality, you have multiple X and then all the X are silenced. So this has been shown using the RNA fish assay. RNA fish is fluorescence in situ hybridizations. Okay, so if it's an important assay used to track RNA molecule in these cells. So using this assay, they identified here the one bare body. This is a link RNA specific fish. So you design a probe for link RNA and then you can identify. Yeah, you see this is a region which indicates ZIST. It's a pretty big regions on the near to the centromeric regions on the X chromosomes. That is X chromosome inactivation center. So ZIST is an important player in the X chromosome inactivations, and then it codes on the own X chromosomes that is defined as a cis acting, and then it recruits a lot of epigenetic machinery and to generate the bare bodies. How does it exactly does? So there is a two transcript need to be balancedly produced from the both chromosomes, active and inactive chromosomes. So the inactive chromosomes produces ZIST. Active chromosome produces T6. T6 nothing but just read reversely, T-S-I-X. Okay, so it's produced from the opposite directions from the other chromosomes, active chromosome. Okay, so the active chromosome, we have the two X chromosome, right? Active chromosome also produces the link RNA that is called T6, and the inactive chromosomes produce link RNA that is ZIST. So the common feature, the inactive chromosomes will have increased expression of the ZIST, decreased T6. You see the you will understand this architecture. The link RNA, this is an inactive chromosomes, it's produced and then T6 absent. And when this is an active chromosome, this is absent and then this is produced on this chromosomes. Basically, the T6 produced from here and then ZIST is produced from here. The T6's name is given because from the opposite direction it has been synthesized. How does it mediate gene silencing? So the ZIST has a many sub RNA molecules, link RNA molecule. One of the RNA molecule is Rep A. So it's a part of the ZIST RNA. This RIP-A, it has a very important functions in targeting the PRC2 protein. PRC2 protein, you all know, it is a polycomb repressive complex protein which carries the enhancer Z homolog 2 proteins, methyltransferase domains. So, the primary methylation inactivation starts with the production of these link cyst RNA, and this RNA, it's spliced out and produces RNA called REP-A, and then this REP-A has an high affinity towards the PRC2, and then the PRC2 are decorated to the whole chromosomes. That's what happened in this case. So, the PRC2 are targeted to the whole inactive chromosomes, which introduces the histone 3 lysine 27 ME3 marks. So, this is the first epigenetic marks being brought into the inactive X chromosome inactivation. The next DNA methylation comes to make it bare body, completely repressed all the regions. So, the cyst transcript is very important, and that there is a REP-A is a cyst also has, but is a spliced form of cyst is a REP-A which has a high affinity to the PRC complex. Whereas the T6, it has a decoy functions. See how it ensures this is always active, because it does not produce cyst, but how it ensures they are always active, because it doesn't allow the recruitment of the PRC2, because the T6 also bind to these PRC2, but it takes away. That's called decoying, I explained, right? It binds and sequesters, similar to the citrulline protein what I explained. So, the T6 RNA bind to the PRC2, and then this is called decoy and not available for the repressions. So, how the active chromosomes maintain always active? Because of the T6 expressions. The T6 is RNA produced from these X chromosome inactivation center from the opposite directions, and which helps to active chromosomes always maintain as an active. Whereas the inactive chromosomes make sure it's repressed, and that's a specific RNA called REP-A. It's a part of the spliced form of the cyst transcript, and which recruits PRC2 complex first, and then introduces the histone 3 lysine 27 trimethylation mark, repressive epigenetic modifications, further DNA methylation mark, and make sure it is a complete. So, they keep in mind every cell divisions, okay, this has to undergo. I mean, that same chromosomes need to get maintained as a repressed. And initially it gets selected either father, mother. So, subsequent cell, when the cell divides, all actively dividing cell has to undergo these mechanisms. It cannot switch the activation inactivation chromosomes. Right. So, this is what in a summarizing what I explained so far, where the REP is a very important link RNA. Okay, it is a part of these cyst only, but it has higher affinity for the PRC2. You see, the few of the knockdown studies or conditional knockout studies in the hematopoietic stem cell for a cyst, you see, it's very important, because born alive few of the animals, but die in a very short time. That means the cyst transcript expression is very important for the normal development in the female baby. And there are a lot of abnormalities when they delete the hematopoietic stem cells. Yeah, so this is the from selection to the from initial selection for inactivation to the fully differentiation, and this is the condensed form of the chromosomes where the cyst transcript is being highly expressed. This is the another example which I explained in the introductory class. The mosaic in logic, here also the mechanism same, the cyst transcript, PRC targeting, everything same, but you have this differential color just due to this mosaicness, because you have color coding gene present in the X chromosomes. So, and that's because of random activations you always have this color. So, this the color coding this kind of brown and black color cat, it is always female cat. This is due to the X chromosome inactivation. This is due to the genetic mosaicsness. Okay, so this is the from the connection to the introduction. It is not always random. For few cases, it is maintained as a father specific. See, the extra embryonic tissues, particularly placenta, only father specific chromosomes are inactivated. Then one of the mammals, which is marsupials, which is a mammalian in most of these Australian countries is present, right? This animal only paternal chromosomes, always every tissues. Very surprisingly, it's said to be detailed mechanism need to be identified here. Only paternal chromosome in this mammal is inactivated. In a mouse or a human, it can be father or mother in the ICM cell, right, in a cell mass. But here, every tissues, every body in the tissue will have the paternal chromosomes in this animal, which is very rare case. In some cases, it has been shown that in basic cell line studies, the inactivation is not complete. Sometimes it can just reactivate. This is very rare cases. Using cell line studies, they are reported. And then sometimes it's reverse, but as of now, the study shows that it's not transmitted to the next generations. Yeah, so this is in connection with the mosaicsness, what I explained. It's a random, because of the randomness, you'll have this mosaic, females are mosaic in connection with the X-linked gene expressions. And then one of the disease called muscular dystrophy is developed due to this X-linked recessive disorder, which is well evidenced with the because of this mosaicsness nature. So this is how the female genetic background can be imagined. So you have this inner cell mass random inactivations, and then that continued. You see throughout the body because of this. In each and every cell, there is a variation of these father or mother specific chromosomes inactivations. You have this mosaicsness. Right. So the common features about these inactive chromosomes, which is always repressed most of the gene except the XIC. XICs need to be always expressed because it need to produce the cis transcript, right? And then it is random, that I already explained. And then these chromosomes are highly heterochromatic, need to be, because that's why it forms a repressive nature, and then it is from a bare body. Then it's mostly compared to many other genes, chromosomes, it's late replicating. It need to completely unpack and then replicate and then modify. Then another important feature, it has a macro H2A variants. None of the other chromosome will have this many, but only this will have the macro H2A, which has the bigger H2A than any other H2A variants. So these are the few features about these X chromosome inactive X chromosomes. But there's still a few questions, very difficult to answer. See, if normal XS female has one chromosome inactivated, right? The Turner syndrome, where they have only one X chromosome, but they do have a symptoms. Why they cannot be normal, which is very difficult to explain, right? The Turner females, they always have only an X chromosome, but they have a severe developmental issues. That means, along with inactivations, there is some mechanisms which is needed for the normal development, which is yet to be discovered. And then in a male, one extra chromosomes is present, right? And in a female, all the X chromosomes, what are the excess is present, got inactivated. In a male, in the Klinefelter syndrome, and then it gets inactivated, but still they develop features like female. But these are things, exact mechanisms, it's not very clear. Particularly, this is very surprising. They have only one chromosome is sufficient for the. That is why you have the dosage compensation. Why do they have a developmental issues, which is not very clear. So what is the connection between these? What is the molecular mechanism happening in active chromosomes? Not yet clear. Okay, so I end using these final slides. In addition to the cyst. These are the other RNA also I explained, R-TAR, R-tip and nest. Few other lncRNA also contributes in epigenetic regulations at various level. So, altogether I want to summarize the lncRNA. It contributes the epigenetic gene silencing by either recruiting the epigenetic machinery or by decoying. I mean, sequester the epigenetic machinery not available for the gene repressions or activations. This is the primary functions. The few other rare example which I explained are through R loop formation and so on. The X-chromosome inactivation is a very famous epigenetic phenomenon where the lncRNA trigger the inactivation mechanisms by recruiting the polycomb repressive complexes and so on. Okay, so with this I'll close the lncRNA chapters because this is a good enough. That means with this class we are completed all three major epigenetic players: DNA methylations, histone modifications, lncRNA. We will see the regulate. I am going to explain very briefly about the regulatory RNAs because this is a newly emerging concept. The microRNA, siRNA, pvRNA. This initially thought it contributes only for the directly regulates RNA expression, but now it has been shown that this can regulate expression by modulating the epigenetic machinery. So, since it's a new field, I'll briefly touch on the part. After that, we will move on to the subject specific: stem cells, plant, and model organisms, and so on. Okay.